This is a video recording of the Muscular System Lab materials and objectives on pages 20 and 21 of your lab packet. You'll find some nice diagrams in chapters 9 and 10 of your textbook. So the first objective asks you to identify 3 types of muscle tissue microscopically and name an organ in which that muscle type is typically located. So when you look at the 3 types of muscle tissue, here we can see this is called skeletal muscle, meaning that it attaches to the bones, and some specific characteristics we see with skeletal muscle tissue is that it is striated. You can see these vertical stripes making up each cell. They're long cylindrical cells. Here's a nice little diagram of an actual skeletal muscle cell, so we can see they're long and cylindrical. They have multiple nuclei just underneath the plasma membrane on the outer edge of the cell. And again, they're striated. They have this striped appearance. So this skeletal muscle tissue is voluntary. We can control it for the most part. It attaches to our bones, and it has multiple nuclei and is striated. Then other type of muscle tissue is cardiac muscle tissue, and this type of muscle tissue is also striated. We can see those vertical stripes here. And it also has these darker structures that we'll learn more about when we study the cardiovascular system, but that's unique to cardiac tissue, are these darker lines here. They're called intercalated discs, but you don't need to worry about that for this next lab exam on the muscular system. But we see that the number of nuclei is reduced. It's typically one nucleus per cell, so that makes this a little different from skeletal muscle, which has multiple nuclei per cell. And we also see the muscle cells themselves are branched, so you can see some open spaces here where there's not continuous bundles of these muscle cells, but they're branched in appearance. You can see here on the bottom, unfortunately this icon is in the way, but if you go to your textbook and look at table 6.1, you'll see that the cells of cardiac muscle tissue are branched. So we find this in the heart. So looking at locations in the body, cardiac muscle is found in the heart, skeletal muscle is found attached to bones, and smooth muscle we find lining the walls of our hollow organs, for example, the bladder, the digestive tract, the airways, upper airways, and the insides of blood vessels. So what we see characteristic of smooth muscle is we have these narrow cells, spindle-shaped cells. They're narrow on each end, and they have one nucleus per cell. They do not have the striations that we see in cardiac and skeletal muscle tissue, so that makes them unique as well. And they are arranged very close together in sheets. So this is smooth muscle, again lining the walls of our hollow organs. So be able to identify these underneath the microscope. There'll be some microscope slides in the study room for you to look at each of these different tissue types, and you definitely want to become familiar with their differences and their similarities. So the next set of objectives is asking us to look at the parts of a skeletal muscle cell. So this is one skeletal muscle cell that we're focusing on microscopically. The plasma membrane of a muscle cell has a unique name. We call it the sarcolemma. So what's peeled away here is the sarcolemma, just the plasma membrane of a muscle cell. And the fluid, the cytoplasm inside of this muscle cell, has another special name, and that's sarcoplasm. So sarcolemma is the plasma membrane, and sarcoplasm is the cytoplasm inside that muscle cell. And again, in skeletal muscle cell, we see multiple nuclei. So here's a nucleus here, here's one here, and here's another one over here. So these are the nuclei. They help control the functioning of the skeletal muscle cell, but they're near the outside of the cell. We can see that most of the interior of the muscle cell is made up of these tiny contracting units, which are called myofibrils. So this straw-like structure here that makes up a majority of the muscle cell interior is a myofibril. And the myofibrils are made up of smaller units, smaller protein filaments called myofilaments. And the myofilaments are these repeating banded pattern that we see in skeletal muscle tissue. The darker structures, the darker red lines, this is called myosin, myofilament, so the dark red line here, myosin. And these thinner blue lines here, these are the actin myofilaments. So we have these repeating myofilament overlapping structures of myosin and actin. And then if we look at where the actin is actually tethered to the myofibril, it's at these special structures here called Z discs. And you can see they kind of make a Z-shaped pattern here. So from one Z disc to the next Z disc, this length of the myofibril between these two Z-shaped structures excuse me, is called a sarcomere. So from this point to this point, this distance here is called a sarcomere. And this is where the action of muscle contraction occurs, is the interaction between myosin and actin, and we'll find that the distance between these two Z discs actually becomes smaller during muscle contraction as this whole myofibril shortens. But the myofilaments themselves do not shorten. They simply have more of an overlap. And you'll learn about the physiology of muscle contraction in your lecture. So that leads us to objective number 3. Objective number 3 has us look at the neuromuscular junction. And here we can see a neuron, a myelinated, the blue here is the myelinated neuron, and then that neuron ends in axon terminals. Well, if we focus on one axon terminal, we'll find that it sits over the muscle cell membrane, the sarcolemma, here. So if we look at this little square here and blow it up to look at it in a larger view, we can see that this is the end of one axon terminal, and it's very close to the plasma membrane of a muscle cell. So in looking at some structures here, we can see the axon terminal is this large yellow structure, and then we call it the presynaptic terminal because the space between the neuron and the muscle cell, what's shown in gray here, is called the synaptic cleft. So we call this the presynaptic terminal, and then we call the muscle cell membrane just below the motor neuron, we call that the motor end plate. So the motor end plate is simply the muscle cell membrane just below the area of the axon terminal. So all of this here is the motor end plate. And we find that there's channels here on the motor end plate that will help initiate an action potential in this muscle cell when it's stimulated by the motor neuron. So the space between the axon terminal and the muscle cell membrane, which is called the motor end plate, again, this gray area inside between the two, we call the synaptic cleft. And in your lecture, you'll learn a lot about how a motor neuron can stimulate an action potential in a muscle cell. So if we look at a typical muscle, muscles are attached to bones by tendons. So this structure here, this connective tissue, is called a tendon. And the connective tissue that makes up a tendon extends as a thin sheet over the muscle cell itself, and we call this outer layer that covers muscle, we call this the epimysium. So epi meaning on top. Epimysium is the connective tissue covering on the outside of muscle, and when those muscle fibers end, that epimysium forms a band of tissue called a tendon. Now in some cases in the body, it's not in a banded-like structure, but it actually forms a sheet of tissue, which we call an aponeurosis. So on this model of the head here, we can see this sheet of connective tissue, which is still epimysium, but we call it an aponeurosis. So we'll also find this over the abdomen as well. We'll see a sheet of this white connective epimysium, and again called an aponeurosis. So a tendon and an aponeurosis are the same tissue, it's just what they look like in terms of how dense they are in terms of their connection. So if it's short and in a band-like structure, we call it a tendon. If it's wide and in a sheet-like structure, we call that an aponeurosis. But both are made of epimysium. And then looking inside of this muscle tissue, we see that there are muscle cells that are bundled into what we call fascicles. So this is a muscle tissue, and inside of it are fascicles, and in between the fascicles is another type of connective tissue called endomysium. So what we see between the muscle cells here is another connective tissue called endomysium. It's this light pink between these circular structures called endomysium. So that I think would be best at this point now to look at some pictures of some models that you're going to see in lab. This is a model of a muscle cell showing the neuromuscular junction here. We can see the motor neuron and the gray axon terminals. And we see this kind of indented region here below the motor neuron. That would be the motor end plate. It's kind of red and white striped here. They've cut off the axon terminals for us to see the muscle cell membrane. So that would be the motor end plate. So here what we see in beige, this is the sarcolemma. The purple structure here, this would be the nucleus on the outer edge of the cell. And all of these straw-like structures here, these are all myofibrils. So those are all myofibrils inside the muscle cell. And then if we look at a myofibril, we see that banded appearance. So the darker pink parts of this myofibril, shown in dark pink here, those would be the the myosin myofilaments in dark pink. And then the light pink in between the dark pink, that would be the actin. And then the dark black lines you see, that would be the Z-disc. So from one dark black line to the next dark black line would be a sarcomere. There's another picture of another muscle cell model. It's a little newer. This is the neuromuscular junction here. We can see the motor neuron here ending and coming in contact with the muscle cell membrane. So what we see here, this beige color here, is the muscle cell membrane or the motor end plate. What's shown in white here would be the synaptic cleft. And then this yellow structure here would be the motor neuron. So again, the beige kind of wrinkled structure here would be the motor end plate. The white is the synaptic cleft, and the yellow is the motor neuron. And here we can see again, inside the muscle cell are all these myofibrils. And you can see the banded pattern that we see on the myofibrils made up of actin and myosin myofilaments. Not quite as easy to see the actin and myosin in this particular model, so I wouldn't expect you to expect Deke to use this model for showing those actin and myosin myofilaments. It's shown a little bit better in the other model here. Now here we can see on the outside of the muscle cell, so this is plasma membrane, this is sarcolemma, but on the very outside here, this would be the endomysium, because this is what covers the muscle cell and separates them within the muscle fascicle. And you'll talk about fascicles in your lecture. I'm not going to spend time here talking about that, but this is endomysium, covers the muscle cell membrane. All right, so now we're going to go back to my PowerPoint. I'm hoping, if I didn't lose it. All right, so that concludes the microscopic anatomy of the muscular system. We will pick up next time with looking at objective number 5 on page 21.